NEB TV, what's trending in science. Welcome to the first episode of NEB TV. Today, I'm joined by Peter Weigley, one of our staff scientists. Welcome, Peter.
Hi, Dina. Thank you.
And we are talking about synthetic biology.
So for today's episode, I'm going to give you a 1-minute overview of what is synthetic biology. Then we're going to hear from my colleague Breton about the highlights of the SEED Synthetic Biology meeting, which just happened here locally in Boston. And then we're going to round up the episode with some thoughts about where synthetic biology is going. And we're going to be hearing from Deepak Misra and Chris Voigt, who are both working at MIT. To me, synthetic biology is programming with DNA. And by programming, I mean using recombinant nucleic acids to direct biological systems to execute a subset of predetermined outputs based on the computation of multiple inputs. Program behaviors could be simple, such as the expression of a fluorescent reporter gene in response to some environmental toxin, or more complex with sequential stages occurring in temporal sequence, such as an engineered stem cell programmed to adopt one of several predefined cell states based on its position within a tissue into which it's been injected. The engineering mindset for programming biology in this way borrows heavily from analog circuitry, digital logic, but these are really a framework, a shorthand to facilitate the design of increasingly complex systems of gene regulation. Whatever the approach, the material foundation of synthetic biology is the availability of inexpensive pieces of custom chemically synthesized DNA, as well as the enzymatic methods for putting those DNAs together.
So I'm here with Breton Hornblower, one of our product marketing managers. Hey, Breton.
Hey, how's it going, Dina?
Good. Breton recently attended the SEED meeting, which NEB sponsored. Breton, can you tell us what SEED stands for?
Sure, it stands for Synthetic Biology, Engineering, Evolution, and Design.
And what was the most interesting talk that you attended?
I think probably the most interesting talk was Ron Weiss's talk from MIT. So he described encoding biological circuits in RNA molecules rather than DNA. The primary advantages being that you wouldn't have to integrate with the host genome and that you could also achieve some sort of temporal control with the RNA.
Great. Will you be attending SEED next year?
We will. It's going to be in Chicago in June.
Excellent. Thank you.
Thanks.
So I'm Deepak Misra.
My name is Christopher Voigt.
I'm currently a graduate student in biological engineering at MIT.
I'm a professor of biological engineering at the Massachusetts Institute of Technology. So synthetic biology is about the process of genetic engineering.
So it's a catch-all term of just these are novel functions that a cell may do to some extent, but they may actually be things that we've just completely put in all together and we have engineering control over.
And the ambition of the field is to be able to access the natural complexity that we see in the world and not be limited in the types of products that we can use biology to produce.
If we want to build a molecule or if we want to build a new plastic, we'll get an organism that actually grows and produces that molecule, produces that plastic for us to use.
Well, what we're seeing is an incredible increase in the scale of genetic engineering projects. So a few years ago, it was really only possible to modify a couple of genes as part of a project, but now we're looking at major projects that involve dozens of genes with different cellular systems.
If we have to get a new drug treatment because we're sick, we're going to go in and a doctor's going to take some cells from us and actually grow a new little artificial liver, it's as big as like a fist, and test the various drugs on you to make sure it's actually going to be effective and safe. And then we're going to go all the way where maybe one day you do need to get a skin transplant or an organ transplant. And we're going to encode genetic networks into your very own cells that actually augment your body and are able to do just that.
So it's trying to be able to build systems in biology that are larger and more sophisticated than what's been done previously.
So thanks for joining us today, Peter.
My pleasure.
And if you have any suggestions for future episodes, please let us know.